Sorry about that. Well, welcome to Benito's explanations, okay? Well, that is it.
Finally, this channel has received a whopping 100 subscribers.
And Benito's Explanations has gained 200 subscribers.
Oh, dearie me. Hello there, it's the third video in the Evolutionary Ecology series, and to be honest with you, it sounds a bit weird saying that because the title of today's video was the title of my entire last series. Isn't that a bit strange? So it's sensory ecology. So just like last time, we're going to be looking at adaptations in the wild, measuring selection over time, but this time we're going to be focusing on sensory modalities. Okay? Now, a lot of you, I don't know why you wouldn't, but probably a lot of you didn't see my sensory ecology series. So, what is sensory ecology? Well, it's quite a broad research field, but it attempts to understand the relationships of organisms between the ecological environment and how it influences the communication of information. And this isn't just going to be a direct copy of the 15 bloody videos that I made a few months ago. This is looking at sensory ecology from an evolutionary perspective. We're going to be looking at it from a slightly different angle, which we didn't really do before. Okay, so when looking at sensory ecology, we need to understand what kind of information is obtained by an organism, the mechanism by which it's obtained, and how that can be used as useful information to that organism. So we're going to focus first then on vision, visual ecology, probably the most well-studied sense. So this is basically how eyes are adapted to see light in the environment that they're in. And when talking about evolution, we're mostly referring to what are opsin genes. Okay, so opsin genes are genes which code for the pigments in our eye, our photopigments, that allow us to see the different colours. On the retina of our eyes, we have rods and cones. Rod cells have rod opsins, which are just sensitive to light, really, but the cone cells are sensitive to different colours. So in our eyes, we have three types of opsin. One for long wavelengths, medium wavelengths, and short wavelengths of light. And those together allow us to see the entire visible spectrum of the electromagnetic spectrum. So around 400 to 700 nanometers. But obviously the number of opsins that we have is different to other animal species, and these have arisen through gene duplication events. For example, there's two rod opsin genes, RH1 and RH2, and it's thought that when the gnathostomes, so the jawed vertebrates, diverged from the jawless fish, then that was when that gene duplicated to produce two different genes, which were then free to evolve. Okay? If you look into many clades of fish, you see that there's loads of gene duplication events that have happened, which have meant that fish actually have an incredible array of opsins, which means they can see very well into wavelengths that we can't. Actually, when you look at the mammals, our vision isn't very good at all. In fact, we have lost some opsin genes along the way. Lake Baikal is a rift lake which is 25 million years old, and it's pretty deep. And inside that lake, there's loads of endemic species, particularly of cotoid fish, which is surprising. We only have one species of cotoid fish in this country, that's the bullhead or miller's thumb. But in Lake Baikal, there's a huge radiation of these cotoid fish. Now, because this lake is so deep, that means as you go down, the light environment changes drastically. So that means we can look at these cotoid fish, look at their visual systems, and see whether they're adapted to the particular light environment that they're found in. And what wavelengths of light penetrate in the water depends on the clearness of the water. So in really clear water, as light penetrates down, it begins to attenuate, whilst eventually, once you reach a certain depth, you're only left with the blue wavelengths. That's why the sea's blue, of course. But in more murky water, the blue wavelengths attenuate slightly more, and the green wavelengths don't so much. So that leaves you with a more greenish colour. So remember, blue wavelengths are shorter wavelengths than on the opposite end of the spectrum, so long wavelengths. So as you go deeper down, the frequency of LWS, or long wavelength sensitive genes, begins to decrease. And the spectral sensitivity of the individual opsins begins to refine itself. It tends to shift, the spectral sensitivity tends to shift towards a wavelength of around 480 nanometers, which is pretty blue. And this is due to shifts in the amino acids that code for those particular opsins. Now, Baumeister and Hunt constructed a phylogeny of the RH1 gene for the species of cotoid fish in Lake Baikal. And what he found was quite striking. He saw spectral shifts towards those 480 nanometers, and he found that shifts in amino acids can lead to a change in spectral sensitivity of about 10 nanometers, which actually is quite a lot. And these changes in amino acid sequences are occurring in exactly the same places each time. So this is a fantastic example of molecular parallel evolution. We talked about parallel evolution in the first video about what it was. This is as parallel as you can possibly get, because exactly the same patterns of evolution is happening in these different cotoid fish species down to the molecular level. In shallower waters, there's similar convergence has been found for spectral sensitivities at longer wavelengths as well. Now, there are loads of other lakes out there. Probably some of the most famous lakes are in Africa, Lake Tanganyika and Lake Malawi. And they also have some pretty diverse fish populations. So Sugawara et al. In 2005 looked at the cichlid diversity and did pretty much the same study as what was done by Baumeister and Hunt with RH1. And what they found was quite remarkable. Remember, these are cotoid fish, these are cichlid fish, so two completely different related groups of fish, but they found the same spectral shifts with depth in almost exactly the same amino acid positions. So not only is this an example of parallel evolution, but it's also an example of convergent evolution, because you've got two completely different fish taxa converging on the same spectral sensitivities, which makes them suited to particular light environments. Now, it's all very well known that the genes are there, but it's also useful to look at gene expression of these genes. And it was found in another study that this was very much related to ecology. So by doing these expression analyses, you can see that each species of fish has a completely different expression pattern of these opsin genes. So each of them is seeing in a slightly different way because they each have a different ecological niche. What is quite curious is that things like fish which feed on benthic invertebrates don't seem to be able to see in the UV wavelengths as well. But we're not really sure why that is yet. Okay, so that's vision then. What is there to say about hearing? Obviously hearing is very important to many organisms. And the main case study we're going to focus on is a study on this particular gene called prestin, which is a motor protein, and it's located on the outer hair cells of the cochlea in your ear. Okay, so we've got it, but it's particularly highly expressed in animals which echolocate. And because of this, it's undergone some slight changes at the molecular level. Now, the most famous echolocators of all, without doubt, are the bats. Now remember, we can split the bats into two groups. There's the Microchiroptera, which make up most bat species that we see, but there's also the Megachiroptera. These are the fruit bats, and these don't actually echolocate. These are the really big bats that often you see flying about in the daytime, visually searching for food, which is usually fruit. Okay. Now actually there's also been speculation that echolocation in bats has evolved more than once. But regardless of that, if you just take the Prestin gene, the Microchiroptera certainly forms a monophyletic group. So there's definitely strong convergence if it isn't a monophyletic group. Okay. So here's a phylogeny then. And what I've drawn at the bottom here are the cetaceans. Now the cetaceans are also famous for echolocating, the dolphins and the whales. Okay, so what this guy called Lee did in 2008, he took the Prestin gene and constructed a phylogeny just from that. And what you find is that this phylogeny isn't actually what you see if you just take the Prestin gene into account. Remarkably, whales and dolphins are shown to be more related to the Microchiroptera than the Megachiroptera are to the Microchiroptera. So basically, if you just take the Prestin gene into account, dolphins are more related to bats than bats are to fruit bats. So therefore there's been pretty strong convergent evolution here. And it's not just Prestin as well. Parker et al. In 2013 looked at more genes, and he showed that more of these genes were undergoing convergence. So in this study, 2,000 sensory genes were taken, and of those 2,000, 200 of those, when constructing a phylogeny, those phylogenies were consistent with convergent evolution rather than just following the species tree. Okay, so that's interesting. We'll have a whole video on phylogenies later in the series, by the way, so you'll have that to look forward to. Now, olfaction. Olfaction's a bit of a weird one. I talked about in the sensory ecology series that it's probably our oldest sense. And there's loads of genes associated with olfaction. Hayden et al. In 2010 looked at 8 of these olfactory gene families and looked within mammals the number of genes within each family, within each species. And what they found was, as you'd expect, the expression levels of these different olfactory genes were correlated to the type of habitat that these mammals were living in. So for example, bats, which live in an aerial habitat, had similar gene expression within the similar families of these olfactory genes. Okay, so there are pretty neat and interesting examples of evolutionary convergence on sensory modalities. But how did that all come about? What are the mechanisms behind it? Well, the guy who really devised the main central theory to the evolution of sensory traits was Endler again, John Endler, in 1992, and he devised what's now known as the sensory drive hypothesis. And it basically says that the environment drives strong selection on local populations, and in doing so it's maximising conspicuous against background noise. So that means organisms evolve so closely with the environment that they're in that, just like how we talked about last time with the suicide king, once put in a different environment they don't do so well. And this means there's the potential for divergence, geographical divergence of signalling receptors traits if those two environments are different. Okay? So this, as a result, imposes selection on the sensory systems, and those changes in the sensory systems will affect the source of information that you're trying to perceive. So then you basically get this coevolution. And this is particularly relevant in sexual selection because it will affect mate choice criteria, which will then affect, well, usually in males, what they display to attract females. So there's this constant coevolution going on between signalers and receivers all the time. So that's the basic theory. How do you test it then? Well, this guy called Bauman in 2001 did a study on sticklebacks. Now, sticklebacks, we've talked about before, the males have bright red chests, which are an attempt to attract loads of females in when they make a nest. 6 populations from 6 different light environments were chosen for this study, and these different environments were marked on how clear the water was, basically. So they went from turbid to really, really crystal clear water. And it was shown, as you'd expect from similar studies that we've talked about before, that in clearer water, the males are redder, they're more red. And that's because red stands out much more in clear water, because in more murky water you get more red wavelengths penetrating through, less red wavelengths attenuate. So when the visibility spectrum of the water is red shifted anyway, there's less of an advantage for you as a stickleback to be red. Okay, plus producing all that red pigment is quite costly to produce as well. Okay, so that's the first interesting bit of evidence. The second piece of evidence was that females are more sensitive to red in these clear water conditions compared to the murky water ones. And this was done by calculating the log detection threshold, but we don't need to worry about that. Okay, so you've got the first bit. So males are more red in clear environments. Females prefer red in clear environments. What we need to do now is to establish the link between the two, and that's exactly what was done. It was shown that females prefer red males in clear environments. So there's clear coevolution occurring here between males and females, basically summarising the entire other sensory drive hypothesis. And it's important to show that this preference exists, because there could just be a sensory bias occurring here. Yeah? Just because females are more sensitive to red in clear water environments doesn't necessarily mean that they prefer red. Okay, so it's good to do these behavioural experiments. Now, more work on the sensory drive hypothesis has led people to speculate that this may actually lead to speciation. Now, speciation is going to be the topic of our next video, so we're going to have a lot to look forward to there. But we're just going to do this study by Tobias et al. In 2010. And what he did, he looked at 33 pairs of Amazonian bird. Okay, so each of these pairs, each bird within the pair is closely related to the other member in the pair. Okay, so that's why they're pairs. But what's different about them is that they live in different habitats. So one member of the pair lives in a sort of terra firma forest habitat, whilst the other one lives in bamboo forest. Now, these are quite different environments, really, but they occur quite close to one another, which makes this a very good natural experimental setup. Yeah, we're finding loads of these now. It's just like how we talked about last time with the guppies and the Damia riverside. Yeah? So these habitats differ in understory density, visibility, number of trees, that sort of thing. Also, in bamboo forests, high frequency sounds can't travel as easily, whilst terra firma forests distort fast-paced songs. There's loads of little constraints here, and it was found that in bamboo forests, calls were of a lower pitch, they were faster, shorter, and had fewer notes. So therefore, there seems to be a strong divergence in these song characteristics. But could this be down to other factors? Could there be some correlated evolution? For example, these differences could be due to differences in beak size, okay, which might be selected for through some other factor, and because of that the calls sound different. Well, actually, the scientists looked into that and they actually found no, that's not the case. So it's thought that these differences in call properties could have possibly have led to the speciation event between these two populations. Okay, so that's quite interesting, because obviously calls by birds are very important in mate choice. Right, that's it. I'll see you next time. I'm talking about speciation next time. Toodle pip.